All right, greetings, welcome all. What's the plan for today? I wanted to pay my debts from Friday when I didn't quite finish in the allotted time. So I wanted to talk about feature extraction for semantic segmentation. You should know this keyword by now. We will use this elastik program for that. And then Alberto will walk you through exercise number 1. For those interested, he will give you a refresher in Python. And for those who don't have an exercise partner yet, there will be matchmaking in the exercises. And before I start, could I please have a show of hands of those who still need an exercise partner? Good. So we've been talking about convolution filters at length, and we've also been talking about semantic segmentation. So assigning one out of several possible classes to each pixel in an image. On Friday, I showed you the image of a street scene, and the typical task would be to say this pixel is road, this pixel is sidewalk, this pixel is vegetation. And that is important for path planning. You like to drive on roads, but not on trees and so on. Yeah? So that's a typical semantic segmentation task. My lab, we're in particular working a lot with biological images or biomedical images. Now, I want to show you a few slides, and I've also copied them into the presentation. A slide I gave to bioimage analysis crowd a few years back, and I've been preaching the gospel that machine learning is good. This was slightly before it became a great fashion. By now it doesn't need much preaching anymore. Everyone is convinced that this is a very useful tool in bioimage analysis and beyond. So at its heart, what supervised machine learning is about, that you give examples as opposed to trying to invent pipelines by hand and adjust parameters or thresholds by hand. So this is an image I like a lot. You might have seen it on the screen of the computer. We are looking at a rat cerebellum here, so a tiny part of the rat brain. And let's assume, for example, that we want to just segment foreground from background. So what we need is a few features. And if I select 2 features, for example, this could be maybe the brightness of a pixel and the difference of brightness between this pixel and the neighboring pixel, for example, that would be 2 features. Then I can embed each and every pixel in a 2-dimensional feature space. So if I have 1,000,000 pixels here, I get 1,000,000 points, and this is just a scatter plot for each pixel of what is its first feature versus what is its second feature. Then I can give a few annotations in the image domain. And what matters here is only the color, not the size of these blobs. I just wanted to show that I can mark a few pixels shown here in red as background, and a few other pixels shown here in green as foreground. And of course, I know what the features are for those pixels. So I can map these features to my feature space. Or I can map these labels to my feature space. Then in this feature space, I now need to come up with a decision rule. And I could, for example, just try a linear decision rule that separates the red from the green, or I could try a nonlinear decision rule such as this one. I've just drawn this by hand. And then I can apply this decision rule to all pixels. So in feature space, this is what it would look like. So everything on the left of the decision rule has now been marked green. Everything on the right-hand side of the decision rule has now been marked red. And these predicted values I can now map back to the feature space, and will obtain something like this. And you see that, it does a decent job of foreground background segmentation, but not perfect. You see there are a few things which are marked as background, even though they should not be. And well, the reason is that, and I've kept this on purpose, such supervised machine learning algorithms, they only ever know what you have taught them. So if we go back to the original image, you see that those parts here, yes, we understand that this is a part of brain, but they are actually very dark, very similar to this background here. And so probably one would then want to give additional labels. And if we're really unlucky, then the features that we selected are not appropriate for discriminating between foreground and background. Or in other words, if the features are not appropriate, then I will not have a clear separation between the labeled pixels of the one class and the labeled pixels of the other class. And then my classifier will just overfit, or it will give bad predictions. And if the features are inadequate, the only thing I can do is add more, hopefully more informative features. I've given you another example here. I have a two-dimensional feature space, x1 and x2. I have two categories, and I've marked them here in terms of color. I have the black category and I have the red category. And well, if I ask you to come up with a decision rule, so some rule that will separate the black points from the red points, you will have a hard time finding a decision rule, right? There's no clear discernible trend here. However, this is because, well, these features are uninformative, and if you then add an informative feature, so previously we had x1 and x2, and now we have added an informative feature x3, then we see that suddenly the two classes do become discernible, and it will be possible to learn a rule that helps me discriminate my categories. So having informative features is really, really essential in these types of learning applications. And what we are doing here, that's a very generally applicable scheme that we take some image, it may be gray value image, it may be color image, then we send this through some filter bank. So it means we extract all kinds of new images that you see on the right-hand side. And these new images can result from a mere convolution, so can result from a linear operation acting on the original image. Or there are also important nonlinear filters that I may use to create these new images. So, in some sense, you can say that I did not really create new information, because after all, I just started out from this original image, but I now have created a few additional images that all encode some information about the local neighborhood. So previously, at a single pixel, the only information I had at the single pixel level was just its gray value. And the gray value alone, or the color alone, is often not enough to discern your classes of interest. So remember the street view example. If something is red, well, this could be the jacket of a person, or it could be the red fender of a car, or it could be the gas tank of a red motorcycle. So just the color alone is not going to be very informative. We need some more information, we need some more non-local information. And in a way, a lot of what computer vision is about is to aggregate non-local information to make meaningful decisions locally. And in this example here, well, we have used linear and nonlinear filters to come up with a whole stack of responses. So now at each single pixel, I no longer have a mere single number, a scalar number encoding the gray value, but now I have an entire vector that says something about the local neighborhood of that pixel. And so coming back to this metaphor here, maybe the gray value alone is not sufficiently informative, but these higher dimensional representations may then be enough to discern the classes of interest. Right? So finding or using the right features, there is a little bit of experience that goes into that. And once we have created our feature space, well, okay, let's say prior to neural networks, this was really all about experience. One of the beauties of deep learning is that we can learn which features are useful. However, even the use of deep neural nets still requires a bit of experience these days, and what this community is trying to do is to make it simpler and simpler for the person, so to make less and less experience required in order to successfully train a machine learning method here. Good, so now once we have settled on a feature space, there is still the question of what classifier do I use? And I previously said we could have used a linear rule or we could have used a nonlinear rule, but how do you do that? And the very simplest classifier that exists is the 1 nearest neighbor classifier. So here I have observations in a two-dimensional feature space. Each point is one observation, and I can read off what is its feature number 2 and its feature number 1 for each and every observation. I can then partition my feature space according to which is the closest observation. This is a Voronoi tessellation. And then I can remember the labels that I had. And if I now get a query, such as here, so I have measured this value for feature 1 and I've measured that value for feature 2. Which class is it? The red class or the blue class? Well, then the 1 nearest neighbor rule asks in which Voronoi region do I lie? What is the label of my nearest neighbor? Hence the name 1 nearest neighbor classifier, and that happens to be from the red class. And then I would predict red for this query here, and I think that's quite reasonable. If I had asked you, what would you predict? I think you would have suggested red for this particular point in feature space here. Okay. Now this is just one out of a huge number of different classifiers, and people who followed the machine learning course last semester, they know close to a dozen of different classifiers. If you have plenty of training data and plenty of compute power, then these days you want to use a deep neural network. If you have little training data and little compute power, then you would like to use something like boosting or random forest. And indeed, random forest is what we're going to use here and in the exercise. So this was about machine learning. And as I said at the time, machine learning is good, but interactive machine learning is better. Why? Because if you keep the human out of the loop, then you need to supply lots of training labels in advance, but you don't know yet which are the easy and which are the hard examples. And if you have no training labels to start with, and if your machine learning method is sufficiently fast, then it can save you a lot of human effort. If you give a few labels, you make a prediction, you see where it goes wrong, you add more labels, and you iterate this process. And usually that means that with the same number of labels, you get much higher quality, or you need fewer labels to reach the same quality of result. And that is also helpful in playing with the set of features that you extract from your data when trying to solve a particular problem. All right, and I want to show you that in a demo in a moment. But before I do, I would like to ask if you have any questions. Tobias, by the way, your microphone is not switched off, so we hear some occasional noise from your microphone. Questions, anybody? All right. Then I want to show this to you in real life. I will use the Elastic program. We started this in the lab a dozen years or so ago, and when Anna Kreshuk became group leader at EMBL, with her lab, she took responsibility for the program, and I will just run you through this from scratch. So I'm going to start Elastic here. And I will then use, it has a few workflows, but the one I'm going to use is called pixel classification. Actually, what it does is semantic segmentation, and I expect all of you to know by now what semantic segmentation is, but this was geared mainly towards biologists who will usually not know that term, and hence we just called it, this workflow, pixel classification. Good, so I need to create a project, and on this desktop here, I'm creating a project, and then I can load some data. And I have a small collection here of data sets for demoing various properties. And I'm going to use here the background of my computer screen, but I'm just using a gray value image here. And usually, hopefully you will have train and test images. So if you have train and test images, then you would add another image here. But here actually I only have the single image. Then I'm going to feature selection. And you see that we have features here called color, edge, or texture. And you can open these and then read in more detail what these features are. But for now, I'm just going to use color at the smallest scale. So that means pretty much I'm just using the original gray value really at each pixel. And then I can go to training. And you see I have two labels here. And let's say that I want to segment only these cells here. So I'm painting this cell here in yellow. And I'm painting this in blue. And I press on live update and I get an update. All right. Now it also colored all of these things here yellow, but let's say that I really only want to pick up the cells. So I can do blue here, but well, that has really suppressed the other cells very strongly. Let me highlight label 1 a bit more strongly. You see it looks like, you know, very noisy, my prediction. I can threshold it by clicking here on segmentation, and yeah, it is not great. And well, I want to argue that the reason here is that we have not used adequate features. So just the gray value alone is not sufficiently informative to allow you to pull out these nuclei. Now we can try and do a little bit better by selecting more features. So for example, I'm now using what's called here edge features. Let's say I do this at a few scales. And well, I'm also going to compute texture features, let's say at a few scales. And we can now browse these features. So this was with a little bit smoothing, that is with a little bit more smoothing, and you see that actually this brings out the nuclei quite nicely, and this is with even more smoothing. But we see that yes, it enhances the nuclei, but also it enhances this region where foreground meets background. This here is gradient magnitude, structure tensor, a nonlinear feature. And what's nice is that you see this one here, it brought out the nuclei, but it also brought out this foreground-background transition. Here the structure tensor, on the other hand, it only emphasizes this, but not that. So that gives me some hope that taken together, actually these features might be useful. And let's try this now. You still see my annotations from before. So I had blue annotations here and yellow annotations there. And I'm pressing on live update. And now, well, I do get the nuclei, but I still am picking up a lot of this stuff, the foreground-background transition. And actually you see that I don't have any labels yet for foreground-background transition. So I'm now going to label a little bit of this blue. And yeah, it takes away most of that, while the nuclei are sort of surviving. And now I can try and refine this here until hopefully I get something decent. But I think you get the overall idea. And yeah, not perfect, but much better, I would argue, than the super noisy result we had beforehand. And now, obviously, before I now go away and have solved the problem, I would need a separate test image to see whether what I've done here, or many test images to see whether what I've done here actually generalizes. But what I try to emphasize for you now was this importance of having informative features. So please remember these images here. Here it's impossible to discern the black class from the red class, and here it is possible. And so much of image analysis computer vision is about coming up or learning the right features for a particular problem. Now this program here, it does not use the one nearest neighbor classifier, but random forest, so that's an ensemble of decision trees. That's a very nicely behaved algorithm when you have few training data and when you need a pretty fast turnaround. All right, let me ask again, do you have any questions at this point? Not at all? Okay. By the way, when you do load more than one image, then somewhere here there's a button which lets you toggle between these different images so that you can have a look or flip between your training images and your test images. And then I wanted to point you to a particular resource. So if you want to learn about some of the other tricks or some of the other workflows that Elastic has, or if you want to learn about other tools for bioimage analysis, there is a nice seminar series that has just started. And so actually, pretty much as we speak, there is a talk about segmentation with an algorithm called Stardist, which is a really nice algorithm for segmenting convex objects. Then tomorrow there will be one about a tool for quantitative pathology, and then there will be one about MorphoLibJ, morphological image processing, and then there will be one about the other workflows in Elastic. And this new BIAS is a really nice training organization, and some of the tools that you see here, I think they're actually very well, I think they might really be useful for you one day, because all of these tools are quite potent, but they are made with wet lab people in mind. So it means they all have in common that they're good at something without requiring any programming or without requiring a huge amount of training. And all of these tools are actually pretty interesting to well, go the first steps towards solving a particular problem, and then often you need a little bit of post-processing or hand coding to finalize the result. So everything featured here, I think, is actually really useful and interesting. All right. This is all that I wanted to say. Last chance, any questions from you to me? There is something in the chat, and I will now pause the recording. If I find the right button.